Start recording. No pointer? How am I going to flip?
Where will you put the pointer?
I'm going to so I will use this to move. Okay, let's do Bismillah ar-Rahman ar-Rahim. And then what are we going to talk about? First, as we always do, it's very important to link between the past and the future. And so I'm going to start with an introduction, which is a revision of concepts you already know, and then we will go through the three. Now, the introduction is some sort of a revision. I didn't also find the time to make portfolio slides, but we can ask about this. Describe types of neurons with examples. Very quickly, very quickly. What are the three types of neurons? Pseudo-unipolar, because I'm interested in this in particular. Pseudo-unipolar neurons which are found in dorsal root ganglia and cranial nerve nuclei. Which cranial nerve nuclei?
Sensitive.
Are you sure?
Don't say that.
Yeah?
Don't say that.
Pseudo-unipolar neurons are found in mixed cranial. We are going to confirm this in this lecture because we are not going to find them in this lecture. So this lecture is about sensory training. What do we expect then? What do we expect? We will find one of them. Sorry, there is one pseudo-unipolar neuron, no problem. But what do we expect to find? Bipolar neurons. Pseudo-unipolar neurons in dorsal root ganglia and mixed cranial nerve plus one of the special sensory cranial nerves. And then we have bipolar neurons, and we see them in special sensations. Nerve serving special sensation, particularly the olfactory and the optic, but not the vestibular. And only there. We don't find these bipolar neurons elsewhere. We find them elsewhere, but also in the same sense organs. So we find them in the sense organs, in the nerve serving these sense organs, but nowhere else. Multipolar neurons, we find them everywhere, but particularly in the central nervous system. This is the first question. These are the neurons, these are the multipolar, these are the bipolar, and these are the pseudo-unipolar. I just remind you that still some of the books mention unipolar. But this is a pseudo-unipolar neuron, it's a sensory neuron, and they mention some unipolar neurons as being motor neurons found in the central nervous system. And then we have multipolar neurons. Okay. And sometimes you will find other types. So there is pseudo-unipolar, this is wrong. And this is unipolar, and this is called anaxonic. Anaxonic, no axon, just dendrites. One concept I also ask you to look it up. Pseudo-unipolar neurons, do they have dendrites? And are the peripheral processes of pseudo-unipolar neurons, are they myelinated? You studied sensation, pain sensation, blah blah, see fibers, optic transmission, myelination, does this and that. Are sensory? Do sensory nerves have dendrites or axons? And are they myelinated or not? And are dendrites myelinated? Dendrites? No. Okay. What we have in sensory nerves in general, whether cranial or spinal, are they axons or dendrites? I give you.
In the nerve, it's axis.
Huh?
In the nerve, it's axis.
In the nerve, what do you mean in the nerve? Yes. Whenever you go and read about nerves, they talk about axons. Even if they are receiving information and bringing it towards the central nervous system, they are dendritic in nature, but they are peripheral axons because they are myelin. This is a very important piece of information, but these are bipolar neurons. Now, in the sensory pathways, and you already started the physiology of sensory pathways. Point number one, I keep repeating it. Difference between tract and pathway. The tract is one neuron starting from A to B, one neuron. While pathway, the tract, sorry, I got the tract is one neuron starting from A to B. While pathways, series of modality specific tracts. They serve only one modality. So today when we will speak about the olfactory pathway, the visual pathway, because it's a series of modality specific neurons. So this is very important, a series of modality specific neurons. Now, what is a receptor neuron? Very important. You studied histology. Have you studied? You have a lecture? You had a lecture on receptors? So I asked in physiology and in the past, long past maybe, in histology. In histology, huh? Disappeared? خلاص? Okay, 2 minutes on this because it is relevant. What are receptors? What is the nature of receptors? What are they? Huh? This is functionally, yes. They respond to mechanical, chemical, electric
There are modifications of
stimuli and
there are modifications of the ends of sensory neurons.
Receptors
Receptors are modifications of the end of the sensory neuron.
No, just a minute. Keep the thought because now I am completing the sensory definition. Then we come to the structural definition. The sensory definition is 2 parts. They receive. What do they receive? Mechanical changes, photo, energy, mechanical energy, chemical energy, all of that is received like this. What do they do?
Converted into electrical impulses.
Yes, what do you call that? Physiological sleep transduction. They do transduction. They need something to pick from here the electric impulse and take it to the center. Okay, what are they structurally?
The end of sensory neurons which are modified to receive information.
Yes, modified to receive. How modified? And are all of them modified?
Some are free nowadays.
So some are free nerve endings, some are modified nerve endings, and some are
Nerve ending in a cell, something like that.
huh?
Nerve ending is in a cell, specialized cell.
Yes, this is the third type. They can be free nerve endings, modified nerve endings, or modified cells. The cells themselves are modified to become receptors. Can you give me examples of the third type because it is very relevant? Huh? Taste buds. But are taste buds cells?
No. They are a group.
They are a group. Inside the group itself we have taste neurons, taste receptor neurons. Because we have other cells in the taste bud.
Yes.
So isn't this one taste receptor neurons? Where else? All the special cells. All the special cells. Yes. And you know the names of the cells in the different sense organs? Taste sensory neurons or taste receptor neurons, and we have olfactory neurons because they are receptor and they are everything. We will see that in a minute. And then we have special modified neurons in the retina. Do you remember the structure of the retina? You studied it?
This is
I
ILEI.
will come with the special.
Remember, huh?
No, it will come with the special senses.
Yeah, not
Not towards the.
After we talk about the nerves,
yes.
how can I
How
talk
come?
about the nerves?
How come?
Anyhow, there are modified neurons known as the rods and the cones. Because some of them look like that, rods, some of them look like that, cones. And in the hearing apparatus, no, because there are two. We have auditory hair cells and we have vestibular hair cells. These are modified neurons, neuroepithelium. Remember the neuroepithelium, where does it come from? From the neural crest cells. Remember when we said that we have a fold and then the fold is the tube and then at the junction the cells migrate to form the peripheral nervous system. So they make these cells. Neuroepithelium, we call them the neuroepithelial cells or the neural crest cells. So they develop into receptor neurons. They are not receptors, they are receptor neurons found in relation to the special sense organs. We will see them when we go and continue. You remember these, huh? First, second, third. This is typical in spinal nerves and typical in mixed nerves. But as you will notice today, they are quite atypical in purely sensory cranial nerves. We will see whether we have first order, second order, third order. What follows the third? Where are the third order neurons? Thalamus. What follows? Huh? Cortical neurons. So we have cortical neurons, and these cortical neurons are found where? Where are they found? But where?
In
Any areas?
One, two, and the sensory
That's somatosensory, thank you very much, yes. We don't have one for vision, one for hearing,
Yeah,
one
there is
for... What do we call the first one following the thalamus? The area containing the first cortical neurons, the primary. But we have more cortical neurons in secondary, tertiary, and in association integrative area. So we have a series of cortical neurons. So this is important. We will see about this when we start discussing individual nerves. Okay, this is typical in spinal cord, spinal nerves, first order, second order, third order neurons. But when it comes to the sense organs, it's a bit different. I'm not going to go into the details, but you know, this is a gustative cortex related to taste, olfactory cortex. We have different cortices related to these special senses. Okay. Another question. No, this is just the listing. The listing we just mentioned. The free nerve endings, modified nerve endings, and then modified neurons. And there they are. These are the types. Free, modified. These are the modified ones, and these are the free nerve endings. And these are the bipolar neurons. The modified neurons, we already mentioned them, we just go through them very quickly. Neural crest cells give rise to peripheral nervous system here and there and everywhere. And then especially we have pseudosensory neurons, we already mentioned, in spinal nerve, in ganglia. Ganglionic bipolar neurons are receptor neurons. We have ganglionic bipolar neurons and we have receptor neurons. When we come to identifying them as first order, second order, and third order, there is confusion. Are we going to start counting from the special cells which are modified to become receptors? Do we call the receptor a neuron because it's a modified neuron? Is it a first order neuron? Or shall we consider the one next to it picking the impulse and taking it centrally as the first order? Most books ignore the receptor neuron as one of the series and just talk about it as if it's a receptor and then start counting. This is the first order, this is the second order, this is the third order, and we are going to do it. When we come to olfactory, optic, vestibular, cochlear, we will say, okay, this is the receptor neuron, followed by this is the first order. Then the second, if they go to the thalamus, then it's the third order neuron. Okay, so receptor neurons we already mentioned in the eye, ear, nose, and in the mouth. I said the mouth because taste buds are not in the tongue only. You have taste buds in the palate, you have taste buds even in the larynx. They are transducers, they transduce the energy into electric energy, the different types of energy, and then you see, the signal is then picked by the first order, not the second order. This is a mistake. Not the second. The second neuron, not the second order neuron. Huh? Which is a mistake, sorry. The second neuron, not the second order neuron. It is the second neuron in the series, but it is the first order neuron. Now, you see why this is in red? The dendritic axon. The dendritic axon of a bipolar neuron. We have a bipolar neuron which has got two axons. One of them is dendritic in nature, physiologically speaking. Even structurally, it has got more nerve endings. It's a dendritic axon, and we have another one which is the axon, which goes central. So the electric signal is picked up by this dendritic axon. Example, as you said, taste buds. But as you can see in the detailed diagram, there are taste buds and there are other cells. So these are the modified neurons. And these are the dendritic axons. The dendritic axons coming from another cell. If it is taste is carried through which nerves? Yes? Glossopharyngeal, yes. Hypoglossal is a motor nerve.
Yes.
So sensory is because it was
Taste. Trigeminal nerve. The mandibular division of the trigeminal nerve. The lingual branch of the mandibular division of the trigeminal nerve, which in fact carries taste from the anterior 2/3 of it, while the glossopharyngeal and just the posterior. So, tongue is a representation because I told you earlier, taste is also found in the pharynx, found in the larynx. We have taste buds elsewhere, not only in the tongue, but this is just a representation where we find the taste buds, and this is how they look like. And then this is a magnification. Now the second cells. Look at these cells, because when we start discussing the olfactory, I want to go very quickly because the olfactory has got only one neuron doing everything. It is the receptor neuron, it is the first order, it is everything, and directly goes to the cortex. But through a number of cells, they are part of the cortex. They are an extension of the cortex, but they are cortical neurons found in an extension of the cortex called olfactory bulb. these neurons are the receptor neurons, and they are the first order neurons at the same time. So this is a big difference, and we will address this issue more later. The third one are the rods and cones found in the retina. They are found in the retina. These are specialized, modified neuroepithelium to become receptor. These are the receptor neurons of the. They are found as part of a layer of the retina. In the retina. Now, this is the retina. Light comes from this side, and the receptors are here. Can you imagine?
Yeah.
Light comes and passes across the whole thickness of the retina. Then it is detected. The receptor neurons are not on the surface of the retina, they are at the external end of the retina, and light enters the eyeball from within. It goes through the iris, then goes to the retina, and then passes through the retina till it hits this pigment epithelium and reflects back. Then it is detected by these rods and cones. Then they come back. The signal transduction takes place here. There are some bipolar neurons, which are the first order neurons, and there are the second order neurons, which give rise to the optic nerve. And then the optic nerve goes to the thalamus. Does it go to the thalamus? Yes, it does, but it goes directly to the cortex. Okay? I will see that in a little while. The fourth modified epithelium are the hair cells. We have auditory hair cells and we have vestibular hair cells. We don't have time to go into the details. It's a very complicated issue. It needs time, but we will discuss them with vestibulocochlear. Okay, just a reminder, and each time I bring an illustration with an error so that you remember that the internet and books are full of scientific errors. What is wrong with this illustration? This is a bipolar cell.
It should be dendritic axon.
Yes, salam alaikum. Thank you very much. They are not dendrites. They are terminal arborizations of this dendritic axon. And this is what is also wrong?
There is no myelin.
But there are some sensory neurons which are non-myelinated. Example, and remember it from now. By the way, unmyelinated neurons typically found in the autonomic nervous system. But the autonomic nervous system is a motor system, not sensory nervous. So this you just need to remember this. Now these are the four types here, but these are the touch. These are the special senses, but the touch and pain and all of that, free nerve endings, means the muscles, you know, the encapsulated ones. But these are the modified neurons that we talked about. Okay, olfactory nerve. We already started talking about it. How many neurons? One. Only one. But this is not the only difference between olfactory and other neurons or other pathways. There are other differences. It's very unique. The olfactory system is very unique, or olfactory pathway. The first order neuron lies at the body surface. No other first order neuron is found right on the surface. This one is exposed to the surface. And because it is exposed to the surface, let me finish with this. The axons of the first neuron enter the cortex directly. No second order neuron, no third order neuron, no directly to the cortex. This is another difference. The neurons, because they are found in the surface, they are different from other neurons. Other neurons rarely regenerate. But these are every couple of continuous turnover, every few days. The olfactory epithelium is changed, the olfactory receptor neurons are changed. So this is a big difference. And then the pathway to the highest cortical centers is ipsilateral. What is that?
On the same side.
Huh?
On the same side.
On the same side. They do not cross. But the others, they deposit, they cross, and all of that, but this is different. They do not cross. Excellent. You have a problem here or here, same side, same side. So these are differences between olfactory pathways and.
I have to excuse myself.
Yeah, no, sorry. Okay. I forgot to change the text and colors because I think there is an image covering this. Now, the first neurosensory cells, the receptor neurons, the first one, the neuron of the olfactory system, found in the olfactory epithelium. And the olfactory epithelium is found in the lateral wall, medial wall, and the roof of the upper part of the nasal cavity. Why do you think that's the case? Why up there and not on the walls, not on the floor? Yes, but not. This is the only, not. This is not the only reason.
Protection.
Huh? Protection, yes, but this is not the main reason. It is dealing with odors, and odors are vapors, and vapors go up. So we wait for them up there. They go into the nasal cavity, they will arise, so they are captured by the olfactory epithelium. So it's in the medial and lateral walls. Now the dendrites or the dendritic ends, dendritic ends of these bipolar neurons reach the surface and form what is known as dendritic bulbs, the vesicles, dendritic bulbs or vesicles. And these bulbs, they have very long cilia. Yeah, it's bulb have long cilia up to 100 micrometers, which radiate into the covering film of mucus. طبعا عارفين mucous membrane is covered with a thin film of mucus. Embedded in that mucus are the cilia. So we have a dendritic end with a bulb, and in that bulb there are cilia. These cilia are embedded into the mucus film. Now, very fine filaments, slow conducting, unmyelinated axons ascend through foramina in what is known as the cribriform plate of the ethmoid bone. And so we don't have an olfactory nerve, we have olfactory nerves. Because the foramina are 12 to 20. And in each foramen there is a bundle of axons running through the opening. So we have 12 to 20 olfactory nerves on each side. These are olfactory nerves. And these olfactory nerves go through this cribriform plate, 12 to 20, and then about 25 million axons form these around 20 olfactory nerves, or bundle or phyla. In one end, the projections are called cilia, and on the other end, the projections are called phylum. And these phyla make bundles, and the bundles are olfactory nerves that go through the. Now, 25 million. This is why, although olfaction or smell in human is not as important as it is in animals, but it is still a very big capability. You smell something, you know what it is, although there are different odors. So it's highly sophisticated. It's sophisticated because of the number of actions and because of something physiology lecture will tell you more about it. There's something called the glomerulus in the olfactory bulb, a very highly sophisticated structure. The smell is a highly sophisticated sensation. Just a summary, these are the cells, neuroepithelial cells. On this side we have the bulb, and in the bulb we have the cilia. These are the axons, and they make phyla, and the phyla make bundles, and bundles are phyla, and they go up to the bulb. The bulb is an extension of the cerebral cortex. This is where we find them. This is the bulb. And this is a magnification of this. This is the bulb, and these are the second, actually the cortical neurons, both sides of the nasal cavity. And as you can see, these are bundles of axons, not just one axon, surrounded by Schwann cells. Okay, I don't want to go into details about the bulb. You can read about it. The most important structure is that it contains something called the mitral cell. So the mitral cell, is it the second order neuron?
No, there's no
This is the
second
first cortical neuron. This is the first cortical neuron. So I need to skip it. There it is. These are the cells, mitral cells. And I told you I don't want to go into the details because, as you can see, there are different things here. There are other cells in the bulb. When you discuss olfaction, you go into the structural details of the bulb. And now this is the bulb. It's an extension of the cortex. There it is. And this is called the olfactory tract, which is formed of the axons of the mitral cells in the bulb. They're axons. They go back and they go to the cortex, which is called the olfactory cortex. I'm also not going to go into this. Show you here. This is the bulb. This is the tract. Tract has got two types of fibers. Some of them go to the cortex directly, olfactory cortex. Some cross to the other side. It's a decussation. You will learn about it with olfaction. And there, when you study in detail what happens in relation to cortical activities, you will know that olfaction is very much associated with the limbic system. What is the function of the limbic system? Emotions. So emotions, olfaction. When you have a diagram like this, all of these are elements of the limbic system. And as you can see, it is connected to all of them. Olfaction from the bulb directly connected to part of the system. This limbic system is the olfactory cortex, which is the second cortical neuron. Second cortical. Very quickly, do you think olfaction is important?
Yes.
What for? What for?
For example, it's important for taste. Because if you can't smell, you will not be able to taste it.
Okay, like better muscle.
Sometimes because what if you can smell the toxins and then you just don't go somewhere and then you
You are hovering around what I am
like for example, if you're in a place and that has like poison smell around it, if you can't smell it, you wouldn't be able to know.
But is it only poisons?
I mean any smell.
No, it's burning.
What
Burning,
is
yeah.
supposed to smell?
It's alarming.
It's a warning system.
Warning.
Yes.
It's a warning system. When you come to eat something and
It smells like it.
you throw it in the dustbin.
Yeah.
So this is a warning. If something is burning, if gas is leaking, it's a warning system. So it is important for survival of what else needed for survival of the individual is eating. Now, eating is called a desire, and desires need motivation. Imagine that when you have the flu and you don't taste, don't sense the flavor of food, you don't eat. You have anorexia because you need motivation. So it's also a survival system. So it's survival by avoiding danger and by also eating. And it's also important for the survival of the human race. So it is also important for reproduction. Then, olfaction is important, but to prove to you that it is not considered very important, what is loss of vision in English? Blindness. What is loss of vision بالعربي in Arabic? Right. What is loss of hearing? Deafness in Arabic? أطرش أو الصمت. What is loss of smell in English? What is loss of smell in Arabic? خلاص خلينا من الإنجليزي. So people don't think it's important. They didn't even bother to give lots of smell a meaning, neither in English nor in Arabic. But it is, and because it is important for survival of the individual and of the race, you need to be aware of when there is a problem. So it's important, stimulation and excitement and enjoyment, especially in relation to human desires, necessary for survival and perpetuation of the race itself, eating and reproduction. As an early warning system, it's believed that its role is less essential than in lower vertebrates. Already said that. So loss of smell has got only a technical name. It's a technical name, anosmia. Anosmia. Is not considered a serious problem unless it is an indication of a more serious one. No synonym. It has got no. Now, if you have bilateral anosmia, can result from disease of the nasal mucosa. Well, it's bilateral. But when a patient has got unilateral anosmia, you have to be more careful in dealing with it because it's most likely due to a tumor or a fracture or something on one side, or sepsis or whatever. Now, unilateral can be an indication of a tumor. Unilateral anosmia is tested. How do we test it? We ask the patient to close their eyes and we let them smell something and tell us what it is. But it has to be common odors. Beans, something usual. But don't use something which is a stimulant because it will be detected by the trigeminal nerve rather than the lower part. It will cause irritation, and so you give them coffee, camphor, etc. with the eyes shut, and then don't use irritants because irritants stimulate the trigeminal fibers, so don't use them. Finally, there is something called olfactory hallucinations. They start smelling odors which are not there, and this is usually with temporal lobe epilepsy or lesions. Lesions. Time is up and maybe over. Yes. We will try to adjust that. We will continue from here forward and finish with these two great nerves very quickly. I will try to be as quick as I can, but I don't think I can be more quick than this.
Suggest more lectures because we will end up not understanding the rest.
Doctor, remember when they gave us like we had like 2 lectures and when they used to go to the workshop, so ask for those.
Yeah, I can. Yes, I will. And by the way, remember that I didn't finish.
You didn't finish functional columns for master.
Yes. So, write them down and remind me. We will ask for more sessions.
Thank you, Doctor. This is